Welcome, dear students. I'm back after a long time. I was actually busy, so unable to upload upload a lecture on my channel. So we will continue where we stopped, and we were discussing in the last lecture. We were discussing the metabolism of carbohydrates, and we were discussing the process of glycolysis. Glycolysis. As you know, glycolysis, it is a combination of two words. Glyco means sugar and lysis means breakdown. So the breakdown of sugar molecule is called glycolysis. Glycolysis is of two types, Arabic and anaerobic. Arabic, when the normal process of glycolysis is Arabic, in which the end product is pyruvate. And anaerobic condition, when we have lack of oxygen, the end product becomes lactate. So there is only a difference that in case of anaerobic, we obtain the end product lactate, and in Arabic and a normal glycolysis, we obtain pyruvate. So glycolysis basically has two phases. It has two phases. One is the preparatory phase, and one is the payoff phase. Payoff phase. Preparatory phase, we also called it investment phase. As the name indicated, investment phase. So we invest a small amount of energy, and in a payoff phase, we obtained a large amount of energy. So by investing a small energy, we obtained a high energy. So that is why we called it that is an investment phase, where we are investing the energy in the form of ATPs, and in a payoff phase, we are taking the profit in the form of ATPs. Now, how this process occurs? So, dear students, the process glycolysis, we will discuss glycolysis with glucose. In the first step, this glucose, we have taken a lot of carbohydrates. Mostly we take starch, the polysaccharides. These polysaccharides are digested, absorbed, and goes into the cell. And when we take polysaccharides, they are converted into monosaccharides like glucose. This glucose goes inside the cell, where it is first converted into glucose 6 phosphate. Phosphorylation of glucose occurs. This is the first step of glycolysis. Means this is how the cell locks the glucose molecule, because glucose, when goes inside the cell, glucose has the tendency that it can comes out of the cell. So what cell will do with that glucose? This glucose is phosphorylated, and in this case, one molecule of ATP is converted into ADP. Adenosine triphosphate is converted into adenosine diphosphate, and one phosphate is incorporated at position number 6 of glucose, and it becomes glucose 6 phosphate. Now in the next step, this glucose 6 phosphate by a reversible reaction is converted into fructose 6 phosphate, which is the isomer of glucose 6 phosphate, and the enzyme responsible here is isomerase. Here we have the enzyme. In case when glucose is converted into glucose 6 phosphate, the enzyme is hexokinase. Kinases are responsible for phosphorylation. So isomerase convert glucose 6 phosphate into fructose 6 phosphate. This fructose 6 phosphate in the next step is converted into fructose 1,6 bisphosphate. 1,6 bisphosphate. The enzyme responsible here is fructose phosphofructokinase 1. PFK1, phosphofructokinase 1. As I told you, kinases are responsible for phosphorylation. We have already a phosphate group at position number 6. Now, at position number 1 of this molecule, one more phosphate group is attached, and it becomes fructose 1,6 bisphosphate. In this case, again, one molecule of ATP is converted into ADP. and one phosphate which is released is incorporated here and it becomes fructose 1,6 bisphosphate. Now fructose 1,6 bis, this is where the preparatory phase ends. This is the investment phase where we invested two ATPs. Here you can see one ATP is invested here, one ATP is invested here. So this is the preparatory phase. This is the preparatory phase or investment phase. Now after this, the second phase, payoff phase or the lysis phase starts. Now when the lysis starts, this fructose 1,6 phosphate, which is a 6 carbon molecule, is converted into two molecules of three carbon molecules, that is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Both are the three carbon molecules. So the six carbon molecule is break down into two molecules which are composed of three carbons, glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Okay, so the fructose 1,6 bisphosphate is cleaved into two molecules or three carbon molecules, that is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. The enzyme responsible is aldolase. The enzyme here is aldolase. Now this dihydroxyacetone phosphate is isomerized into glyceraldehyde 3 phosphate because in glycolysis this glyceraldehyde 3 phosphate continues to run. So when it converts, the enzyme responsible is isomerase. Now this glyceraldehyde 3 phosphate is then converted into 1,3 bisphosphoglycerate, 1,3 bisphosphoglycerate, phosphoglycerate. This is a reversible reaction, and the enzyme responsible here is this glyceraldehyde 3 phosphate is converted into 1,3 bisphosphoglycerate. The enzyme responsible is dehydrogenase. The enzyme responsible here is dehydrogenase, and in this reaction, one molecule of NAD NAD plus is reduced to NADH plus H plus. And you know, when one molecule of NAD is reduced to NADH plus, it produces three ATPs. So here we have one molecule of NAD is converted into NADH plus is reduced to this. Now in the next reaction, this 1,3 bisphosphoglycerate is converted into phosphoglycerate is converted into 3 phosphoglycerate. One molecule of ADP is converted into ATP. Enzyme responsible is phosphoglycerate. And the enzyme responsible is kinase. Phosphoglycerate kinase is a complete name. This 3 phosphoglycerate then is converted into 2 phosphoglycerate. 2 phosphoglycerate. This 2-phosphoglycerate then is converted into phosphoenol pyruvate, and finally it is converted into pyruvate. Pyruvate. And this is the payoff phase. From here, it is the payoff phase. Payoff phase. As I told you, glycolysis is divided into two phases, preparatory phase and the payoff phase. This is the preparatory phase or the investment phase where we are investing two molecules of ATP and we are here we obtain the ATPs. Now we will discuss with the structures. As you know, we have started with the molecule of glucose. So in detail, if we go into the detail with the structures, this is the structure of glucose. As you know, glucose is a 6 carbon molecule. 1, 2, 3, 4, 5, and 6. This is a carbonyl group. So glucose, this is the aldose sugar. This is the aldo group. It is a 6 carbon. This is D-glucose. So we started from glucose. This glucose molecule first converts into glucose 6 phosphate. This is carbon number 1, 2, 3, 4, 5, and 6. So at position number 6, carbon number 6 phosphate group is introduced and it becomes 1, 2, 3, 4, 5, 6 CH2OP. This is the phosphate group. Remaining structure remains the same. This is glucose 6 phosphate. In this, one molecule of ATP is converted into ADP. The one phosphate which is released is incorporated at position number 6 and converts glucose into glucose 6 phosphate. Enzyme responsible is hexokinase. And you can also have this hexokinase needs a cofactor, that is a magnesium. So in books you may find that magnesium is also written over here. So it is a cofactor which is required by the hexokinase to work at optimum. Now glucose 6 phosphate in the next reaction is converted into, by a reversible reaction, is converted into fructose 6 phosphate, which is an isomer of glucose 6 phosphate. As you know, fructose is also a 6 carbon sugar, 1, 2, 3, 4, 5, and 6. We have CH2OP, OH, H, H, OH, OH, H, double bond O, CH2OH. This is the keto group. The difference between glucose and fructose is glucose is an aldose sugar having an aldehyde group, and fructose is a keto sugar. Both have the same empirical formula, they are the isomers of each other. So the enzyme responsible here is isomerase. Now this fructose 6 phosphate is then converted into, by again it is converted into fructose 1,6 bisphosphate, 1, 2, 3, 4, 5, and 6 CH2OP. This is OH, H, H, OH, OH, H, double bond O, CH2OP. Now at carbon number 1, another phosphate group is introduced, and the enzyme responsible is phosphofructokinase 1. This is an important step. This is a regulatory step when we will discuss the regulation of glycolysis. So we will discuss the importance of this enzyme, phosphofructokinase 1. In this, the enzyme responsible here, this is here one molecule of ATP again. One molecule of ATP is converted into ADP. The one phosphate which is liberated is introduced here. So it becomes fructose 1,6 bisphosphate. Phosphate. This is where the preparatory phase of glycolysis ends. Now this fructose 1,6 bisphosphate uh, becomes enters into the payoff phase and the lysis starts. Up till now, you can see we have started from 6 carbon glucose, having a 6 carbon, glucose 6 phosphate, 6 carbons, fructose 6 phosphate, 6 carbon molecule, and fructose 1,6 bisphosphate, 6 carbon molecule. Now, this 6 carbon molecules in the next step is cleaved into two molecules, cleaved into two molecules of 3 carbon molecule, that is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. The enzyme, this is a reversible reaction. The enzyme responsible is aldolase. This dihydroxyacetone phosphate converts into glyceraldehyde 3 phosphate by the action of enzyme isomerase. Now, what we have seen that this 6 carbon molecule fructose 1,6 bisphosphate is break down into two molecules of 3 carbon, that is glyceraldehyde 3 phosphate. Now we obtain two molecules of glyceraldehyde 3 phosphate which runs parallel to give us two molecules of pyruvate. Now glyceraldehyde 3 phosphate, which is a 3 carbon molecule, 1 2 3 CH2OP OH, and it is having an aldehyde group. This is a molecule of glyceraldehyde 3 phosphate. Now we have two molecules of glyceraldehyde 3 phosphate, so they runs parallel. This glyceraldehyde 3 phosphate in the next reaction is converted into 1,3 bisphosphoglycerate. 1,3 bisphosphoglycerate. 1 2 3 CH2OP OH, and this is 1 3, so CH2OP. Now we have two phosphate groups, so this phosphate basically comes from, in this reaction, one molecule of NAD. This is a reversible reaction. One molecule of NAD, nicotinamide adenine dinucleotide, is reduced to NADH plus H plus. So 1,3 bisphosphoglycerate, the enzyme responsible here is dehydrogenase. Dehydrogenase. The complete name is the glyceraldehyde phosphoglycerate dehydrogenase is the complete name of this enzyme. You can also write dehydrogenase, it is enough at this level. So this 1,3 bisphosphoglycerate is converted into 3-phosphoglycerate. 3-phosphoglycerate. 3-phosphoglycerate means one phosphate group is liberated here, and in this molecule, one molecule of ADP is converted into ATP. From where this one phosphate comes, one phosphate is liberated by this 1,3 bisphosphoglycerate, which is incorporated here in ADP, which converts ADP into ATP. So here you can see we are again obtaining the energy. Now, in this, the enzyme responsible is kinase. The complete name is phosphoglycerate kinase. This 3-phosphoglycerate in the next step, okay, this glyceraldehyde 3-phosphate is converted into 1,3 bisphosphoglycerate by the action of enzyme dehydrogenase, and in this one molecule of NAD is reduced to NADH plus. This is a molecule of 1,3 bisphosphoglycerate. This 1,3 bisphosphoglycerate in the next reaction is converted into 3-phosphoglycerate. The enzyme responsible is kinase. Here one molecule of ADP is converted into ATP, and one phosphate which is liberated from 1,3 bisphosphoglycerate is incorporated here. One phosphate is incorporated here and ADP is converted into ATP. 2-phosphoglycerate, 2-phosphoglycerate by the action of enzyme enolase is converted into phosphoenol pyruvate, and the structure of phosphoenol pyruvate is C double bond OH. This is CHOP and double bond CH2. This is phosphoenol pyruvate. The enzyme responsible is enolase, and it is an irreversible reaction. This phosphoenol pyruvate finally converts into pyruvate. This is COOH. This is CH3, and it becomes OH. This is pyruvate. And in this reaction, again one molecule of ADP is converted into ATP. And this reaction is again an irreversible reaction. This is a reversible reaction, this is irreversible reaction. So here you can see we have started from glucose, which is a 6-carbon molecule, 6-carbon molecule, and we have ended at 3-carbon molecule pyruvate, two molecules of pyruvate. One is this glyceraldehyde 3-phosphate given one pyruvate, and this glyceraldehyde 3-phosphate gives the second pyruvate. So overall, In glycolysis, we have the end product. This is aerobic glycolysis, where we have the end product, which is this pyruvate. So, in this, how much energy is invested and how much energy is obtained, we will discuss in the energetics of glycolysis. So, the next lecture will be on the energetics of glycolysis. Till now, thank you. If you have any questions, you may ask in comments. Thank you very much.